Hello, everyone. I am Ishtiak Rashid. I'm a PhD student at the Laboratory of Mathematical Biology at Kaido University. Today, I'll be talking to you about my research on machine learning based stratification of asymptomatic carriers of HTLV-1 and exploring candidate biomarkers for identifying high risk carriers of HTLV-1 associated myelopathy, also known as tropical spastic paraparesis. So, what is HTLV? HTLV is a human T lymphotropic virus. This is a human T lymphotropic virus type 1. This is a retrovirus, and it causes two types of disease. One is a type of blood cancer called ATL, and another is a inflammatory disease of the central nervous system called HUM. The way of transmission is through body fluids. It's prevalent in some parts of the world, including Japan, especially in southern region like Okinawa and Kyushu. So what are the challenges? Why it is important to identify high-risk carriers? First, low occurrence rate. Only a few of HTLV-1 carriers develop the disease, and they remain asymptomatic for lifelong. However, additionally, when a rare disease takes 5 years to be detected, HTLV-1 related diseases take more than 7 years. Also, it's not predetermined among who will develop the disease. Clinically, there is still no established method to identify the high-risk people, but PVL, proviral load, has been used. But there are some lack of specificity which makes it more confusing. So this is a proviral genome, 9 kilobase per ROM, and the list of structural, regulatory, and accessory genes. In our previous study using LEAPS assay, we obtained antibody titers, and we evaluated the presence of antibodies among the non-infected controls and HTLV-1 carriers, and we found strong response in GAG antibodies. Usually, the HTLV previous researchers used GAG as a whole, but we found the mature GAG is clipped by the viral protease during assembly, and three mature protein, P15, 19, and 24 are generated, and we obtained strong immune responses against these GAGs. We added PVL and in this study, and like we we prepared our model. So the data set was quite low, and it was non-time series data, and we collected the data from Kumamoto University Hospital. Comparatively, there were higher number of carriers. First, we performed the correlation analysis, and we found that GAC 19 and 24 had a higher correlations. Therefore, based on the BIF score, we removed P19. So I used anomaly detection on carriers. Anomaly detection is commonly used in many types of research. Usually, when data point generate that unusual data points, it results in anomalies. So isolation forest is an unsupervised anomaly detection algorithm. The main idea is the anomalies are few, and therefore they are detected quickly than normal data points. On the left image, to isolate a data point which is in layer, it took 15 splits. On the other hand, it took comparatively less splits to identify an anomaly. So basically, there are three main steps. It identifies how it identifies the anomaly. First, it randomly takes a subset from the data and randomly selects a feature. After that, it splits the data, taking a value within the range of this feature. And the process is recursively repeated until the tree reaches its depth or all the anomalies are identified. So the pathway anomaly score is a measure of how anomalous the data point is. And in scalar implementation, The negative score represents anomalies and positive scores represent inliers. We applied isolation forest on our carrier data, and using a threshold at minus 0.05, we obtained the inverse 95th percentile of the data. So we found 17 anomaly carriers and 247 non-anomalies. Here is an overview of anomaly detection. On the left side, on a PLS plane, we plotted all of our data, including anomaly samples. And on the right side, the contour plot is used to see the distribution of anomaly scores of the carriers. We can get an overview of distribution of anomaly scores from this image. After that, we did some statistical analysis to characterize the anomaly samples in comparison with other samples, other subgroup. So we can see anomaly samples are showed comparatively higher median values than other clinical subgroups. Also, we calculated the Euclidean distance from anomaly subgroup to other subgroups, which showed it's close to harm, relatively smaller distance with harm. So here is a key summary of statistical analysis. Anomaly samples had very significantly differ with the normal samples, and anomaly samples were not found statistically significant with harm, which resembles they act more like harm. And we also found strong, significantly difference of GACP 15 and 24 among the other subgroups. So far, we stratified the asymptomatic classifier into two groups, and after that, we used rest of the data to build a classifier model to further characterize or understand the fate of the anomaly samples. So here, we used all of the samples for training and used only the anomaly samples for test. Using a 1 versus rest classifier, we used four traditional algorithms because the data set is quite low and low number of samples and low features. So based on the AURC score, we selected random forest and we predicted the anomaly samples. Out of 17, 14 samples were predicted as harm, 2 samples were predicted ATL, and 1 sample was predicted as carrier. Later, we performed feature analysis, and interestingly, we found GACP 15 as the most important for anomaly carriers. Also, we found some variations in other subgroups. Finally, we performed individualized disk profile analysis using SHAP for all the 17 samples, and we took the mean value, and we also found that GACP 15 was the most important in identifying high-risk carriers. So here is a summary. First, we stratified the anomaly carrier population. We characterized it using classifier modeling, statistical analysis, and feature analysis. We showed that anomaly samples resembles harm-like characteristics, and P15 and P15, GACP15 and ENV offer significant advantages as a biomarker for identifying harm-like carriers. And P15 might be a target for immunotherapy. So in the future, we want to validate the method using independent data sets. Actually, we have, but due to the ethical, we haven't received the ethical approval yet, so it's still pending. And also, we want to analyze the time series data. Thank you for listening.
Thank you very much for your talk. Any questions or comments?
Thank you for sharing your work. I want to ask that.
Could you ask a question near here?
Yeah. Okay. Thank you for sharing your work. I'm curious to know, like, how you plan to use the results of your work and to help cure this rare disease. And do you see any future work which progresses towards that? Thank you.
I didn't understand the question.
Oh, I just want to know that how do you think your research can help cure this disease?
Okay, so the main problem is the disease occurs only a few percentage of people, like 0.4%. It's really low, and in 95% of like in most of the cases, it remains asymptomatic. So, moreover, it takes 7 to 30 years to be like detected. Like this virus can remain asymptomatic for 7 to 30 years. So you have we cannot apply like PCR or something for 30 years, of course. So we need to apply some other methods. Like antibody titer is really easy. We can collect the blood samples and applying the isolation forest combined with the classifier, we found that if we can detect the anomalies from the carrier samples, they resemble ham-like. At least we can guess the high risk samples who might develop harm. Like remaining asymptomatic for a long time makes it difficult to identify or any kind of therapy because we don't know who will develop the disease and when.